All right, hi everyone, Claire here. I obviously am not feeling well, so I apologize if I am difficult to understand. My voice is kind of shot, but I will do my best to get through this lecture. All right, so we are still on chapter 17, which is covering blood. And we left off on Monday talking about how red blood cells are formed, their functions, and how they're broken down. So the last part of 17.3 in your text is just about some disorders of red blood cells. Okay, so the first one we're going to talk about is anemia, right? And anemia is kind of a broader term for just any time blood has abnormally low oxygen carrying capacities, okay? Obviously, we need oxygen to support a normal metabolism in our body, have our cells functioning properly, being able to use energy, sugar, etc. So if we don't have enough blood, if don't have enough oxygen, if blood can't carry enough oxygen, obviously it's going to cause us a number of different problems. Okay, anemia isn't technically like a disease on its own. It's a sign of a problem rather than a disease itself. Okay, so other diseases, other problems that you have in your body will lead to anemia, but anemia is never like the first, the main problem, if you will. Okay, if you don't have enough oxygen carrying capacity, you will have symptoms such as fatigue, pallor, shortness of breath, and chills. Okay, and there's 3 different groups of anemia based on the cause of it. So the first is just blood loss, right? Obviously, if you lose blood, enough blood, you won't be able to carry as much oxygen in the blood that you have left. It'll lead to anemia. Okay, you can have not enough functional red blood cells being produced, which can be due to a number of different things. One is iron deficiency. If you don't have enough iron, that heme pigment in the middle, you can't have that iron attached to it. And you'll form something called microcytes instead of red blood cells that aren't as functional. And then lack of vitamin B12. B12 is also needed to make blood, just like iron. So if you don't have enough B12, you'll form macrocytes instead. Okay. In both cases, it's just important to know that you're not being able to actually form fully functional red blood cells. You're forming a type of cell that doesn't carry the oxygen and gases like it should. And then the last cause of anemia is that the red blood cells are being destroyed. So there is something called thalassemia, which makes your red blood cells extra fragile. And basically they just break. It's a genetic disease. And as the blood cells are circulating through, they will break and, be destroyed. And then the other one is sickle cell anemia, okay, which again is also a genetic disease that we're going to spend a little bit more time talking about. Okay, so sickle cell anemia. Our cells obviously are just made of different amino acids, right, to build proteins. And if we have a mutation in our hemoglobin molecule, right, which is that heme pigment and the globin protein, if you have a mutation in that, it'll cause an abnormal blood cell. And we call that hemoglobin S, right? That's a mutated hemoglobin. And what is actually wrong with it is there's one amino acid. If we look over here, amino acid 6 normally is a glucine, and in the hemoglobin S molecule is a valine instead. It produces a sort of normal blood cell, but when there's low oxygen levels, that blood cell becomes a crescent shape, right? So obviously the normal red blood cell shape is nice and round, biconcave. In low oxygen levels, the hemoglobin with this mutation becomes that crescent shape. Okay, so anytime oxygen levels are low, so this can be during exercise, it can be maybe if you're at a high elevation, there's not less atmospheric oxygen, whatever it may be, it causes blood cells to turn into this odd crescent shape, which is why it's called sickle cell anemia, right? Kind of looks like a sickle here. Obviously, this misshapen cells are not ideal. Okay, they can rupture easily or break down. They can also block small vessels. So we talked about it before. Some of the vessels throughout our body are just wide enough to carry, to pass through one red blood cell at a time. Okay, so if we have these sickle cell odd shapes trying to go through these blood vessels, it can cause blockages. Okay, which obviously is going to result in poor oxygen delivery and also can result in pain. Okay. We talked about how the cells are recycled. So if they're recycled, they're going through the small vessels through the spleen. And the spleen is responsible for taking these damaged blood cells out, breaking them down into their parts. So this is what will happen to these sickle cells, right? As they're blocking these little vessels, our body's going to think that they are dysfunctional, which they are, and is going to get rid of them. Okay, so we're going to have definitely fewer cells that are able to carry oxygen efficiently, but even fewer red blood cells in general, just because our body's going to be destroying them. Okay, you might ask, why is this so prevalent? Because we still see it quite often, and it's a genetic mutation that continues to get passed along. Okay, and why is that? You'll see in the African malarial belt, so in regions of Africa, and especially places that have high instances of malaria, right? We see this mutation around more often. And that is because there's actually a possible benefit to it, which is that people with this sickle cell mutation don't contract malaria, which is that disease, as often. Okay, malaria kills a lot of people in the African area, about 1 million people each year. Okay, if individuals have 2 copies of the mutated hemoglobin gene, they will have sickle cell anemia, which will be obviously not good, hard on the body. But if they only have 1 copy of the gene, they have a much milder disease, and they actually have a better chance of surviving malaria. Okay, so if you remember from an intro bio class, mutations of genes that get passed along are passed along because they increases an individual's chance for survival. So in areas where malaria is present, the sickle cell gene will be passed along because it increases their chance of survival. Okay, we're not going to see it in the Western countries because we don't really have malaria as a problem, so there's no need for this gene. But in Africa, you do see that this trait, this mutation, even though it can be very deadly if you have 2 copies of the gene, with 1 copy, it's really evolutionarily beneficial for individuals to have this. Okay, so that's why you still see great instances of that. Okay. We mentioned it a little bit earlier, but there is a disease, whatever you want to call it, a condition called polycythemia, which is an abnormal excess of red blood cells, which is going to increase blood viscosity, which makes blood become kind of sluggish and hard, difficult to move through your vasculature. Your hematocrit, or red blood cell count, may exceed over 80%, and the blood volume may double. Okay, we talked about this before, but it's going to increase your blood pressure, right? If you have higher levels of blood cells, your blood pressure is going to increase. And as the viscosity of your blood increases, it's going to increase your risk of stroke as well. Okay, a few different causes for this guy. If you have excessive erythropoietin production, obviously the EPO is responsible for producing for stimulating, the production of red blood cells. So if we have too much erythropoietin released from our kidneys, it's going to increase the amount of red blood cells being made, potentially to a point where we end up with polycythemia. Bone marrow cancer can be a cause of that. And then the second thing, living at a high elevation. Okay, so if you are living in somewhere, let's say in like Colorado or somewhere up in the mountains, there's going to be less atmosphere, which means less oxygen levels up there, right? And you remember that the kind of balance, if we have low oxygen levels in blood, remember that's going to lead to an increase in erythropoietin production in the kidneys. Okay, so if we live at a high elevation, our body is sensing that our blood isn't carrying enough oxygen, and its response to that is just to produce, increase the amount of erythropoietin so we can increase the amount of red blood cells. Okay, so your body doesn't know it's because of a lack or not enough oxygen available. It just thinks that your blood's not doing its job effectively, so it's going to produce more blood cells. Okay, so people who live at a high elevation typically will actually have to go down or go to the doctor and get their blood drawn regularly just to reduce the amount of red blood cells that they have, because they don't want this increased blood pressure, increased risk of stroke, all the time. So if they draw the blood and decrease the amount of red blood cells, then it'll keep them healthier. Okay, I know we mentioned it before, but there's something called blood doping, where athletes can either take an artificial erythropoietin, or they can remove their blood, store it, and reinfuse it before an event to increase oxygen levels for stamina. Okay, so there's a lot of different ways athletes blood dope. There's different chemicals, drugs, whatever you want to call them, that kind of have a similar effect. Obviously, this is not a good practice. It's not healthy. If we increase, our red blood cell count too much, it can really have detrimental effects on the rest of the health of our body. Okay, so that's, like I said before, not something that we want to happen. Okay. All right, let's move on and talk about some leukocytes. So leukocytes are the white blood cells. And that they are the only formed elements that are complete with cell, nuclei, and organelles. Remember we talked about red blood cells are anucleate, which means they got rid of the nucleus, again, to increase surface area and carrying capacity for oxygen. Leukocytes still keep all of their cell organelles, so they have the nuclei, different mitochondria, everything that they need. Okay, they make up less than 1% of the total blood volume, about 4,800 to 10,800 white blood cells per microliter of blood, so not very many. And their function is to defend against different diseases. Okay, we can group them into two major categories. We have granulocytes and agranulocytes. Okay, granulocytes contain these visible granules, and you'll be able to see those, and you should have seen them in lab already. Agranulocytes do not contain granules. Okay, and these granules are going to have a function, which we'll talk about in a little bit. But our granulocytes, you should be able to categorize these. Neutrophils, eosinophils, and basophils are granulocytes, and agranulocytes are lymphocytes and monocytes. Okay, you should know the relative concentrations of each different type of white blood cell. And there's a mnemonic to remember that. Never let monkeys eat bananas. So the first letter tells you the most abundant white blood cell, neutrophil, lymphocyte, monocyte, eosinophil, and basophil is the fewest. Okay, so you should know the concentration of those. Over here shows you that as well. So again, within the leukocytes, the white blood cells, most abundant neutrophils, never lymphocytes, let monkeys, monocytes eat, eosinophils, and basophils have the fewest. Okay? So this is what they all look like. Again, you looked at these in lab. Again, in the granulocytes, you're going to see these little granules. So if I zoom in here on the neutrophils, so these little granules, or in the eosinophil, these little granules, basophils are harder to see, but there's these dark granules there. That's what make those granulocytes. Okay, agranulocytes don't have that. It's a little bit more difficult to see. But we have lymphocytes, which actually the entire nucleus is this red, dark purple part. So the cytoplasm is just this little wedge on the outside here, and they don't have these dark granules like the other ones. Okay, and monocytes, these little dots here are easily confused with granulocytes, but they're not. They're actually organelles within the cell. Okay, so you'll be able to see granulocytes in the granulocytes and no granules in the agranulocytes. You can see the dark staining nuclei in these different types of cells. And the big thing that is how you're able to determine the different types of cells is based on what the nucleus looks like. Okay, so that'll be really helpful for you guys. All right, let's go through each of these really quickly. I know you already talked about these in lab, so we're not going to spend too much time. Neutrophils are a granulocyte, first of all. They have granules in them. They are the most abundant leukocyte, twice as large as red blood cells, so they're pretty big. They have a lobed nuclei where you can see all these little kind of outcrops and lobes on it. And they have antibacterial activity. Okay, so any bacteria that enters the bloodstream, they are going to ingest using phagocytosis, which, if you remember from 207, is the cell basically eats the bacteria. And then they have within their granules different chemicals to break down and kill these bacteria. Okay, so they are antibacteria. Eosinophils are also granulocytes. They're a bit smaller than neutrophils, but they're similar size. They have two lobes to the nuclei connected by a little stalk. So it's kind of difficult to see, but if this is the lobes of the nuclei, they are connected with just this little stalk there. Okay. They have large granules that stain kind of pink. So if you see a pink staining granules in the picture, you know it's an eosinophil. They have antiparasitic activity. So any type of parasites, they are going to swarm a parasite in your blood and then secrete enzymes onto the surface of the parasites to kill them. Okay, so if you have some sort of parasitic infection, you'll see the number of eosinophils increase greatly. Okay, and again, the number of these white blood cells tells us about our health. If you were to take the blood of someone with a parasitic infection, for example, you would see more eosinophils than you would typically see in a healthy individual. And that goes for all of these white blood cells. So for the neutrophils, if someone has a bacterial infection, you're going to see an increased number of neutrophils present. Okay, so the amount of these types of cells are going to vary depending on the health of the individual. Okay. Our next type is basophils, also granulocytes, our last type. They are the rarest. They are very difficult to see. So if you were in lab and you found one of these in the microscope, you are pretty lucky. They have large, coarse cytoplasmic granules, and they produce a pro-inflammatory molecule called histamine. Okay. And you think of histamine, I like to think of it when there is, think of allergens or allergic reactions, right? A lot of our allergy medicine that we take are antihistamines to reduce inflammation. So these basophils produce the histamine that causes inflammation in our body. Okay, so you can think of them, think of like allergies. okay? Their nuclei is difficult to see because of granules, but it's typically a U or S shaped. I'm not even going to try to outline it because I really don't know where it's at in there. Okay, but the dark blue are going to be the basophils. Okay, then moving on to our A granulocytes, we first have our lymphocytes, which is second most numerous. Again, they have that really large nuclei that takes up most of the cell, and then just a really small sliver of this area here would be the cytoplasm. There's a few different types of these lymphocytes. And they're characterized by size. So there's some small ones that are 5 to 8 micrometers, medium 10 to 12, and large 14 to 17 micrometers. Okay. They all do the same thing, so they're just different sizes. And there's also, though, 2 varieties of them. We have our T lymphocytes and our B lymphocytes. They are going to look the exact same to you. They're going to have similar functions. We'll talk about these more when we talk about our immune system. They're named differently because they mature in different areas of our body. So all blood cells, right, are made in our bone marrow. Most of them mature there as well. Lymphocytes, T lymphocytes actually mature in the thymus. Okay, and then the B mature in bone marrow. Okay, so all of the cells, again, are going to be made in bone marrow. But for some reason the lymphocytes are going to move around. Specifically the T lymphocytes are going to move and mature in the thymus instead. Okay, and we'll talk about their functions more in a next chapter. Okay, and then the last leukocyte we need to talk about is the monocytes. Again, they are also an agranulocyte, no grains, even though they kind of look a little bit grainy. They're not. Those are just organelles. They are the largest. They have about an 18 micrometer diameter, so they're pretty big compared to, like, see right here is a red blood cell. They have a lot of cytoplasm that's kind of pale blue, a dark staining U-shaped nucleus there. And they actually have a very cool function. They will leave the blood and differentiate into cells called macrophages, which means big eater. So they are going to be phagocytic cells in in the body, and do a bunch of other stuff. And again, we'll talk about more specifically these functions when we talk about the immune system and the immune response. Okay. All right, moving on. So leukocyte formation. I'm not going to get too in-depth onto this stuff because I don't think it's very important for this level of class. But I wanted to put it here because I know in Dr. Broughton's lecture he does talk about it, so I just wanted to make sure you guys at least had some information on it. There's 2 different lineages, if you will, of blood cells. Okay, so they're all going to arise from those hematopoietic stem cells. Okay, but after that, they're going to split into 2 different lineages. You have the myeloid stem cells and the lymphoid stem cells. Okay, the myeloid lineage has all of your granulocytes, right? Eosinophils, basophils, neutrophils, and also maybe, we don't actually know, the monocytes. Okay, if you look down here, our monocytes are kind of in the middle here. There is controversy whether they're actually from this myeloid stem cell or if they're from the lymphoid stem cell. Okay, so we don't actually know. But yeah, so monocytes are kind of an unknown. And then the lymphoid, I'll change colors here. The lymphoid lineage, right, again, are going to start at that hemopoietic stem cell. And they're going to go down and differentiate into your lymphocytes, so both your B and T lymphocytes. Okay, you should know though for sure that they all are going to stem from this hematopoietic stem cell, which is that same stem cell we talked about when we talked about the red blood cell formation earlier. Okay. All right, and then the last type of formed element that we have are platelets. They are fragments of larger megakaryocytes. So basically, they're just little pieces of cells that break off. And their function is to form a temporary platelet plug that helps seals breaks in blood vessels. Okay, so it's really important to help with sealing damaged blood vessels and making sure that we don't lose too much blood. Okay, they contain several chemicals that are involved in the clotting process. So we'll talk about the clotting process later on, but serotonin, calcium, various enzymes, ADP, platelet-derived growth factor, a bunch of different stuff that is involved in helping the blood clot so we don't lose, too much blood if there's damage. Okay, platelets age really quickly. They're only around, I think, about like 10 days. So they're made very often. Again, they're just these little fragments of the cell called this megakaryocyte that break off to form our platelets. Okay. All right, here is a summary of all the formed elements that we have. Description, the size, the development, and their function. So if you need a little review, this would be a great place to look. It does a really good job of explaining all of that. And then the very last part of our lecture for this chapter is on hematostasis, which is the fast series of reactions for stoppage of bleeding. So when you think of like blood clotting, a scab forming, that is what hematostasis is. Okay, so first thing is it requires clotting factors. I don't like that color. It requires clotting factors and substance released by platelets and injured tissues. Okay, so clotting factors are just different chemicals that are going to be released that are going to help with this cascade, and we'll go through step by step. But there's three main steps. So the first is vascular spasm. Then you have platelet plug formation, and step 3 is coagulation or blood clotting. Okay. So we'll go into detail on all of these. So here they're in that image. But the first step is the vascular spasm. And we'll talk about vessels more when we talk about that chapter, specifically on vessels. But there's smooth muscles in the walls of our blood vessels. So once we get some sort of little cut or damage there, that the smooth muscle is going to contract, causing vasoconstriction, right? Constriction of the blood vessels. Okay, vasoconstriction there. So these vascular spasms are triggered by the injury to the smooth muscle, also by chemicals released by the endothelial cells and platelets, and pain reflexes. Okay, so there's going to be different chemicals released by the cells, endothelial, and by the platelets that are going to cause these contractions to happen, but also our nerve signals of pain are going to cause this spasm to happen. They're most effective in small blood vessels. So if you have a vessel with a smaller diameter, the vascular spasm, the vasoconstriction, is more efficient in stopping blood. In a larger vessel, we can't constrict enough to really stop all of the blood flow. Okay, but it can significantly reduce the blood flow until other mechanisms can kick in. So the whole point of constricting, again, is to stop or reduce the amount of blood flow through the damaged area so that too much blood doesn't leak out. Okay, I am struggling, guys. 1 second, let me have a sip of my coffee. And if you can hear snoring in the background, that's my dog, Stella. She's sleeping. Okay, so step 1, vascular spasm. Step 2 is platelet plug formation. Okay, so we talked about platelets. Their big job is to form these plugs to block stuff from leaving areas of the vessel that are damaged. So I'm going to zoom in here really quick so you can see. Again, if you have some sort of damage in your vessel, the first thing the vessel is going to do is constrict. After that, a bunch of platelets are going to come and form this plug, right, to help stop blood from flowing out of that damaged area. Okay, so what happens is when there's that tear in the vessel wall, it'll expose collagen fibers. Okay, when those collagen fibers are exposed, platelets will stick to them. Okay, plates do not stick to intact vessels because collagen is not exposed. Okay, so if I zoom in back here again, so let's, in this area that is damaged here, you can see these collagen fibers become exposed, and that is what the platelets are going to come and stick to. Okay, so we need the collagen fibers that are exposed for the blood platelets to stick. Once the platelets stick, they become activated, they swell up, become spiked and extra sticky, right, to help form that plug, and they're going to release chemical messengers. So I'm going to switch colors. These chemical messengers are really important. 1-ADP causes more plates to stick and release their contents. Serotonin and thromboxane A2 are going to enhance vascular spasms, so that constriction, and platelet aggregation. So once the platelets attach to these collagen fibers, they become activated, spiked, sticky. They're going to release these chemicals, which is going to cause a positive feedback cycle. Okay, as more platelets stick, they release more chemicals, which is causing more platelets to stick, which releases more chemicals, on and on and on and on, until we have our final platelet plug. Okay, so that is a perfect example of positive feedback. That the initial stimulus is going to increase and increase until we get what we want. Okay. The last step, step 3, is coagulation. So after we have our vasoconstriction to kind of get our blood vessel a little bit smaller, we have our platelet plug to kind of create a temporary block in our damaged vessel. Our third step is coagulation, which is just blood clotting, which is just going to be there to help reinforce the platelet plug with what we call fibrin threads. Okay, so it's just going to be a protein that comes in and creates this kind of mesh. So if we zoom in here, we got to say our platelet plug over that damage. These fibrin threads are just going to come in and kind of create a mesh barrier to help reinforce that plug even more, to ensure that blood isn't going to be leaving. Okay, so it's sealing larger vessel breaks more specifically. Okay, so if we have a small vessel, the platelet plug might be sufficient in blocking the damage. But if we have a very large vessel break, we're going to need coagulation to actually seal off the damage in that vessel. Okay. All coagulation is blood basically is transformed from a liquid into a gel. It gets thicker. And it happens through a series of reactions that use different clotting factors or procoagulants, which are mostly plasma proteins, that are just going to take that blood, make it thicker, and activate the protein fibrin that we need. Okay, and it's going to occur in 3 phases, and I'll tell you right now, it looks like very confusing to learn. There's a lot going on. But as long as you know the main points of each phase, you'll be okay. Okay. So, 3 phases. 1, 2, 3. Each of these will have a slide. And I know there's a lot going on, but it's really not too bad. So basically what's happening in all of these phases are different procoagulants are going to be activated. Okay, so I'm going to zoom in. So these different clotting factors, so clotting factor 12 right there, it will become activated to 12A, right? So anytime there's that little A, that means the active form. I'm not going to ask you step by step what's activating what. I just need to know at the end of each pathway, each phase rather, what is the main thing that's activated. Okay, so we'll go into detail on all of these, but I'll just show you. At the end of each one, you can see a main thing activated. So in this first one, we have prothrombin activator, right? And there's a few different ways that becomes activated, but prothrombin activator is the main thing that happens in phase 1. Phase 2, we end up with thrombin. And then phase 3, our big thing is that we end up with fibrin, right? Which is that thing that is going to help create the mesh to block in our platelet plug. Okay, so let's go into detail on these. I know this is a lot of text. Focus on the important stuff. So phase 1, again, the end result is you have a prothrombin activator. Okay, phase 1 is the most confusing because there's 2 different ways we can get to this. It can be initiated either by what we call the intrinsic or extrinsic pathway. Typically, when this is happening, it's going to be a combination of both of these kind of working together at the same time. Okay, the clotting is activated by tissue damaging events. Right, these damaged tissues and the platelets rushing in are going to release different chemicals or procoagulants that are going to help create this clot. Okay. So the phase 1, again, our big end goal of phase 1 is the formation of prothrombin activator. I'm not going to ask you about this other shit, but there's a bunch of different other factors. These are all the procoagulants. So factor X is what happens at the end of that intrinsic or extrinsic cascade. It complexes with calcium, platelet factor 3, and factor 5, all to form this prothrombin activator. This is the most important thing you need to know from phase 1. Okay, so there's going to be a cascade of events. All of them are going to lead to the activation of prothrombin activator, and that is what I care about, that you know prothrombin activator is at the end of phase 1. Okay, there is a little bit you have to know about the 2 pathways. So again, we have the intrinsic pathway. Intrinsic pathway has all the clotting factors present within the blood. Okay. So when we have damage to our blood vessel, what's going to happen is we have negatively charged surfaces, such as our activated platelets and our collagen, right, that are exposed or activated when we have damage to our vessels. Those are going to initiate the intrinsic pathway. And the intrinsic pathway has everything, all the clotting factors that it needs within the blood. Okay, so nothing else has to be present. We just have damage to the vessel, activated platelets and collagen, and then everything that we need is present. The extrinsic pathway is kind of the opposite. Factors that are needed are located outside of the blood. Okay? Especially, there's one specific factor that is outside of the blood that you're going to need to know, and that is tissue factor, also known as factor 3. But tissue factor is outside of the blood, and it comes in and then will activate that extrinsic pathway. Okay, so that's the difference between the 2 pathways. I don't expect you to know every single step for each of these pathways, as long as you know that they each lead to the same thing. Okay, so this slide kind of shows that. Again, these are all phase 1. The intrinsic pathway, everything you need is in the blood. Extrinsic pathway, we need exposure of tissue factor, which is from outside of the blood. Okay, they both are going to end up doing the same thing though, right? Which is leading to prothrombin activator. Okay, so there's a series of different cascades, different events, things being activated, whatever. In the end, all that really matters is that prothrombin activator is formed and active. Okay. Phase 2 is the pathway to thrombin. Right, so our first phase, we led with prothrombin activator. Phase 2, that prothrombin activator catalyzes the transformation of prothrombin to thrombin. Okay, so at the end of phase 1, when we ended with prothrombin activator, I'm going to call it PTA. PTA at the end of phase 1 can come in and change prothrombin into thrombin. Okay, so phase 2 is real easy, not hard at all, as long as you know it. The prothrombin activator changes prothrombin to an active form of thrombin. And then the last phase is the common pathway to our fibrin mesh. Okay, again, so what we ended up in phase 2 with was thrombin. Thrombin comes in and converts fibrinogen to fibrin. Okay, and fibrin you need to know. So fibrinogen is something that is naturally found in the blood. It's soluble in our blood, so it's kind of just floating through, dissolved. Thrombin converts that soluble fibrinogen into insoluble fibrin, which makes it so it doesn't dissolve in the blood, and it can make this fibrin mesh that we talk about. Okay, so fibrin strands are the structural basis of the clot. It causes the fibrin causes plasma to become like a gel-like trap, catching different formed elements. Okay. So all it's doing, the important thing is fibrin is made from fibrinogen, and it creates this mesh. Okay, so again, in this image on the left there, you can see that mesh is the fibrin mesh that's going to create that trap and help really seal in any damaged area of the vessels. Okay. All right. So this table in your book goes through all of the different clotting factors that we talked about. You do not need to know all of these. I just wanted to put it up here to show you. There's actually 12 clotting factors because clotting factor 5 and 6 are actually the same, which is kind of funny. They thought they were different, but they're not. And one thing I do want you to look at, most of these are made by the liver, right? Which is why earlier we talked about if we had some sort of liver disorder, it can cause a lot of issues with our blood. And we'll talk about some other issues in the last few slides here as well. Okay, but these are all the different clotting factors. Again, there's a whole bunch of them, and their function in that different cascade of clotting is listed there. You don't have to know them as long as you know the main like end goals of each step of the clotting cascade. Okay. All right, last few slides. So there's a few different disorders, either from excessive clotting or deficiency in clotting. So thromboembolic disorders result from undesirable clot formation. So there's too many clots. Okay, the opposite of that is if we end up with some sort of bleeding disorder. That is due to a deficiency in blood clotting. If we don't have enough clots, obviously we will have different bleeding disorders because our vessels can't form these clots, can't seal themselves off if they get some sort of damage. Okay, and there's two different terms you're going to need to know in regards to clotting. So the first is a thrombus. A thrombus is just a name for a blood clot. Okay, these are made if you have, cuts in your vessels, whatever it may be, but also all the time you have little tears, little damage in your blood vessel walls. So these thrombuses are made literally all the time, nonstop, to help repair just little tears that you're gonna see. An embolus is a dislodged blood clot. Okay, and this is not good. It's undesirable because if we have these little clots that are becoming dislodged, they can go through our bloodstream and can lead to different things such as stroke, different blocked vessels, etc., which obviously is not a good thing. So thrombuses are normal. You should see them all the time in your vessels, little clots being made everywhere just to help repair different tissues. If they become an embolus or a dislodged clot, that's when we're going to start seeing issues. Okay. We have factors in our blood called anticoagulants that are there normally and dominate in the blood to inhibit coagulation. Right? We don't want our blood just to coagulate and get thick and sticky and form these clots. Obviously, we don't want that if we don't need that. Okay, so our blood has natural anticoagulants to help stop that, that are overrun if we do need a clot. But we can also be taking anticoagulant drugs. These will be prescribed to prevent undesirable clotting. So examples of these that you might have heard of: aspirin is an anticoagulant. It's used typically to lower heart attacks, but it also is a blood thinner anticoagulant. Heparin is similar to that, used for pre-op or post-op cardiac care. So after some sort of heart instance issue. Also helps prevent venous thrombosis. And then last one, warfarin. Helps reduce the risk of stroke in patients prone to AFib, which is where blood pools in the heart. Okay, so those are just examples of anticoagulant drugs. Again, they're all going to help prevent unnecessary coagulation or gunking up of these blood. All right, so a few bleeding disorders I want to talk about. Again, bleeding disorders is where you don't have enough clotting. So the first one, thrombocytopenia, is you have a deficient number of circulating platelets. You end up with this petechiae, which are these little red dots all over. And basically, it's just widespread hemorrhage. So you have all these little little pools of blood all over due to different tears and stuff in different veins or arteries, and not having enough platelets to form proper platelet plugs. Okay. It can be due to destruction of red bone marrow. So if you are on radiation, different drugs, a number of different things, if you don't have enough red bone marrow making your different blood cells, it will lead to this. And the treatment for this is just transfusion of platelets. If an individual gets platelets back that they need, they can then again form the platelet plugs that they need, so they won't have these little bleeding events. Impaired liver function can also lead to bleeding disorders. If we can't make all of these procoagulants that are needed to cause a blood clot, obviously we're going to have uncontrollable bleeding happen if there's a damage that occurs. Okay, so if there's a lot of different ways that this could happen, we have a lot of different sources, factors, that are made by the liver, right? So if our liver can't produce those, they'll have issues doing their specified function. But the big takeaway is that if we have impaired liver function, for whatever reason, we can't get these procoagulants that we need, our blood won't be able to clot. So if we have damage to our blood vessels, we will see uncontrolled bleeding, which can lead to hemorrhaging, which is obviously not a good thing. Okay, and then the last bleeding disorder is hemophilia, which is just several similar bleeding disorders that are all hereditary. And what you're going to see is that the individual just has prolonged bleeding, especially into joint cavities. And again, it's just genetic, caused by different things. They're all going to be different factor deficiencies. So depending on what factor, clotting factor is missing, you're going to have different results, different types of hemorrhaging or bleeding. But collectively, these genetic bleeding disorders are called hemophilia. Okay, and the very last thing I want to talk about, we're a little bit over time here today. You talked about this in lab, so very quickly, I'm just going to talk about the blood typing. So in section 17.7 of your text, there's a lot more information. We're just going to keep it very simple here. Okay, so obviously there's a different blood groups. You can have blood type AB, B, A, or O. And is dependent on the antigens on the surface of your red blood cells. Okay, so if you have type AB blood, you have A antigens and B antigens on the surface. Okay, if you have B blood, you just have B antigens. If you have A blood, you just have A antigens. And O blood, you have no antigens on the surface. Okay, so antigens are found on the surface. You also have antibodies that are going to be not on the surface. So I think of the antibodies, these are going to be free guys floating through the blood. So I like to think your antibodies are floating through your body, right? They're not on the surface of the blood cells themselves, they're floating through the body. Okay, so if you have type B blood, you have B antigens on the surface, which means you have A antibodies floating through. Okay. Opposite of that, if you have A blood, you have A antigens on the surface, you have B antibodies floating through. So it's going to be the opposite. So type A antigens, B antibodies. Type B antigens, A antibodies. If you have AB blood, you have both A and B antigens, which means you're going to have no antibodies floating through your blood. And then finally, type O, we have no antigens, so you have both A and B antibodies through the blood. Okay, and if we do a test, if you were to take a sample of blood and put either anti-A or anti-B serum on them, you would see coagulation of the blood happening, which looks, you see how it's a little coagulated here. Anti-A is testing for those A antigens on the surface. Anti-B is testing for the B antigens on the surface. So if you have coagulation happening with anti-A and anti-B, you know you're type AB. If you just react with anti-A, you're type A. Just react with anti-B, you're type B. And if you don't react with either of them, you're type O. Okay, so that's how you do a little bit of blood testing. Again, you talked about this in lab. I'm not going to spend too much time on it. I also posted a Khan Academy video for you to take a look at if you are confused about this or have further questions. They do a pretty good job explaining it. All right, that is the last of chapter 17 and the lecture for today. If you have questions, please let me know. I'd be happy to go over any of the stuff on Friday if you're confused. So let me know. All right, bye guys.